So, hello friends, welcome back to my channel. Today we'll talk about the mechanism of actions of the thyroid hormones. Okay, how the thyroid hormones after productions, how they will go within the cells, and what type of the activations we will see within the cells, as what type of the activities will be done by the cells for the stimulations of the thyroid hormone. We'll talk about all these things in details. Okay. So before going in details, I have to mention one thing in terms of the regulations of the thyroid hormone, that is known as low T3 syndrome. Okay, at first let's talk about that thing, then I'll go in details about the mechanisms. Low T3 syndrome. So how can we define the low T3 syndrome? Now try to understand, for whatever the reasons, if we get the catabolic activity, if we see the catabolic activity within the cells that it has been increased. For example, if we see over here under stress, so if we see that under stress or stressful conditions, stressful catabolisms. So whenever we see the catabolisms under stress, or we can say, for example, for the prolonged starvations, okay, for prolonged starvation, or you can say that the surgery, or you can say for the trauma. So whatever the reasons I have written over here, for all these things we will get the catabolic activity within the cells that will be increased. So whenever the catabolic activity that will be increased over here, what type of problem we will see? The productions of the T3 that will be less. Why? See, under all these circumstances, whatever the thing we will see over here, the productions of the T3 that will be less. Why? See, this okay, these conditions, they will inhibit the what 5' mono deiodinase. Now try to remember, I have already explained that this enzyme that will help to convert the T4, this enzyme that will help to convert the T4 into T3. Now whenever we are getting the inhibitions under stress conditions, for whatever the range I have explained over here, okay, if we see this inhibitions of this enzyme, so what less amount of the T4 that will be converted into less amount of the T3. And as we know, that biologically active form of the thyroid hormone, that is the T3, right? That is the T3. So we will get over here, the T3 that will be severely less. So biologically active form of the thyroid hormone that will be less. So within the cells, we will not get the enough BMR, we will not get enough the proper physical development, we will not get enough the mental development. As a result, we will see the conditions that is known as hypothyroidism. So whenever we will get this type of thing, hypothyroidism, especially if we talk in terms of, we can get the mental retardations, or the physical growth that will be not developed properly. Now there are certain kinds of the factors that will stimulate the productions of that thyroid hormone, and there are certain kinds of the factors that will decrease the productions of the thyroid hormone. So let's write down about them. So first I'll talk about over here the factors what stimulate to produce more amount of the T3 and T4. And there are certain types of the factors. For example, I am writing it over here. Factors, okay, decrease the productions of T3 and T4. So what are the factors behind these things? We have to know in details. Now, as I said, the first factor, there is the TSH, right? If we see the the elevations of the TSH, so if we see the elevations of the TSH over here, so as a result, we'll see the more productions of the what T3 and T4. See, try to understand, as I said, whenever the TSH that will be elevated at the time, we will get the more stimulations within the thyroid follicular cells. So whenever more amount of the TSH that will come over here and they will stimulate more, at that time we will see the more productions of the T3 and T4. Now what are the other factors? Okay, let's write it over here. Now, in certain types of the disease, if I say that in terms of, okay, let's over here, thyroid stimulating immunoglobulin, in the autoimmune disorders over here, if we get in terms of the Graves disease over here, if we see over here, so sometimes we will see the activations of these things, the antibody that will be, sometimes we will see the antibody we will get against the thyroid follicular cells. So whenever this immunoglobulin will activate these cells, so we will see there will be more productions of the T3 and T4. Now what are the other factors? Okay, where we will see, for example, if we get, try to understand from here. Now if we get during pregnancy, what do you know? As I said also earlier before, that during pregnancy the total thyroid binding globulin protein level that will be increased. So during if we see the increase, if we see the elevations of the total, thyroid binding globulin protein that is getting elevated, at that time which within the circulations, more amount of the T3 and T4 they will be bounded by what? They will be bound by thyroid binding globulin protein. So when we will see the thyroid binding globulin protein that will be elevated, in which conditions? As I said, during pregnancy we will get, during pregnancy. Why will see during pregnancy? Because during pregnancy we will see the estrogen and the progesterone hormone. Estrogen and progesterone. These hormones, they will stimulate the what? They will stimulate the liver. Okay, they will stimulate the liver. And whenever we will get the stimulation within the liver, at that time this liver they will produce more amount of what? This liver they will produce more amount of thyroid binding globulin proteins. So whenever more amount of the thyroid binding globulin proteins we are getting over here, so more amount of the T3 and T4 will come over here and they will get bound along with the thyroid binding globulin. So these are the factors. And another thing, if we see the women individuals, they are taking the oral pills. At that time also we can get the elevations of the thyroid binding globulin protein. So if the women individuals they are taking the oral contraceptive pill. So these are the conditions we can say shortly, that these are the factors that can stimulate the productions of T3 and T4. And we'll talk about right now the factors that can decrease the productions of the T3 and T4. The first thing we can say over here, if we see the TSH level that is getting decreased. So as a result, definitely we'll see the T3 level that will be decreased, the T4 level that will be also decreased over here. Okay. Now we'll talk about, for example, if we get the iodine deficiency, iodide deficiency, whenever we get the iodide deficiency, as a result we will get what? The T3 and T4 productions that will be also less. Another thing, if we get the iodide level, that is getting increased too much, if we see the severe increase, if we see the increase iodide too much, if we see the productions of the iodide within the body, the amount of the iodide is too much. At that time, whatever the thing I said you before, that whenever the iodide that will be too much, at that time the iodide pump that will be functionless. So whenever we see the elevations of the iodide too much, at that time, as I said you before also, that the iodide pump that will be functionless. As a result, the iodide pump they will be not able to take the iodine within the follicular cells, and this type of the effect that is known as what? Wolff-Chaikoff effect. That is known as Wolff-Chaikoff effect. So what is Wolff-Chaikoff effect? So let's write it over here. Wolff-Chaikoff effect. Try to remember, I said you also before that, for example, during the surgery of the thyroid gland, before the surgery we used to give what? Potassium iodide. So whenever we get the potassium iodide, as a result we will get the breakdown of this compound and we will get the productions of the iodide too much. So whenever the iodide will be too much at that time, within the thyroid follicular cells, for example, this is the iodide pump. This iodide pump they will be functionless. So whenever they will be functionless, they will be not able to take the iodide from the outside of the cells to inside of the cells. So as a result, definitely we will get the productions of the T3 and T4 that will be also less. These are normal things. Okay. Now, I also said the certain kinds of the drugs. Okay, if you say in terms of the anti-thyroid drugs, as I said, there are certain kinds of the drugs. There are what? The iodide pump inhibitor. For example, if we see in terms of the thiocyanate, or you can say the perchlorate. Okay, they will inhibit the iodide pump. So these drugs, so as a result, the iodide, taking within the cells that will be less. So uptaking of the iodide that will be less. As a result, we will get the less productions of the T3 and T4. And there are also certain kinds of the drugs that will inhibit what? That will inhibit the thyroid peroxidase enzyme. So we get the inhibition of thyroid peroxidase enzyme. What are the drugs? They are known as what? Okay, propyl thiouracil, or the derivative of these things that is known as what? Carbimazole. Okay, let's write it over here. Okay, curvy, it's carbimazole, or you can say the methimazole. So this type of the drugs, they will inhibit what? They will inhibit the thyroid peroxidase enzyme. As a result, we will see all the processes, all the processes, most of the processes for the productions of the T3 and T4 that is done by the thyroid peroxidase enzyme. So whenever it will be inhibited by using the propyl thiouracil, or you can say the derivative of the propyl thiouracil in terms of the carbimazole or the methimazole, they will inhibit this enzyme. So definitely the productions of the T3 and T4 that will be dropped. Okay. So these are the conditions we can get. Okay, in these cases, the productions of the T3 and T4 that will be less. And what else we can say over here? Try to understand if we get, okay, if we get the liver cirrhosis, for example, you are getting the liver cirrhosis. So whenever you are getting the liver cirrhosis, at that time, try to understand the liver, they will be not able to produce the more amount of the thyroid binding globulin protein. So we'll see over here the thyroid binding globulin protein that will be less secreted by the liver. So whenever this protein will be less secreted, at that time, try to understand from here, at that time we'll get what? We'll get the less binding of the T3 and T4 within the circulation. So ultimately within the circulations, the T3 and T4 that will be less. So another thing we can say over here, if we get, for example, the nephrotic syndrome. So if you see the protein that is going out from the body, for example, in terms of the nephrotic syndrome, during nephrotic syndrome, I have a separate video, you can watch it over there. So I said you there that we'll get the proteinuria. So around 3.5 gram per day, every day we'll get this much protein that will be excreted out. So whenever we get the protein excretions, definitely the less amount of the protein that will be available within the circulations to bind the T3 and T4 along with them. So ultimately we will get also the dropping of T3 and T4 in the nephrotic syndrome. So these are the conditions when you will see, the less amount of the T3 and T4, these are the factors. Now exactly now I will come in details about how the cells, are getting activated by the T3 and what type of the receptors are over there and what type of functions we will see within the cells at the cellular level. Okay. Now we are going to talk right now at the cellular level. So what type of thing we will see. Now for that thing, okay, let me erase some part, guys. Okay, erasing this. Now what do you know normally that from the what? From the within the hypothalamus we know there is the presence of the some neurons within the paraventricular space area, we can say also. From here we will get the releasing of the TRH. So TRH will be released by these neurons, and this TRH that will reach from the neurons to the what? Anterior pituitary by the what? This is known as what? Hypothalamic hypophyseal portal system. So by this system we will see the TRH that will reach to the anterior what? Pituitary cells, and I already explained how the mechanisms will be over there to produce the TSH. So the TSH that will be produced, now the TSH will come where within the TSH that will reach where within the thyroid follicular cells, they will activate it and they will produce the T3 and T4. Now whenever we are getting the productions of the T3 and T4, what did I say? You try to remember this T3 and T4 that will be taken by the circulations. So we will see that will be taken up by within the circulations, and as I said that the liver, let me just make a barrier for it. Okay. Now as I said you try to understand the liver. Okay. The liver will produce what? The liver they will produce. See. Anyway, just try to think it liver anyway. Okay. Now liver they will produce the what? Different types of the proteins. For example, the thyroid binding globulin proteins, thyroid binding prealbumin, thyroid binding albumin, and we will see that produced T3 and T4. For example, is the T3 and T4 they are coming over here. They will get bound along with the thyroid binding globulin proteins. Maybe the albumin T3 and T4, maybe with the prealbumin, and with the albumin and the prealbumin, less amount of the what? Less amount of the T3 and T4 will get bound. Now, as I said also before, the affinity for the T4, along with this binding globulin protein is much more greater than the T3. So here we will get the affinity for the T4. Affinity for the T4 that will be greater than the T3, where within the thyroid binding globulin protein. Now, whenever we are seeing that they are getting bounded along with these things, so we will get 99% of the T3 and T4 they will be bound along with these structures, and only 1% of the thyroid hormone that will be in the free form. 1% that will be in the free form. Now how they are going within the cells and how what are the actions we are getting over here, we'll talk in details right now. Okay, for example, I'm just enlarging these portions, circulatory systems. Okay, now here we got what? Here we already got the T3 and T4. Here we already got the T3 and T4. As we know that the T4 that is not biologically active, so whatever the thing we'll see, there is a presence of one kind of enzyme that is known as what? 5' mono what? Deiodinase. They will remove one more iodine from the T4 and the T4 that will be converted into T3. Okay. So for example, we are getting the T4. T4 is already what is the active form. It is able to do the work. But whatever the T4, this T4 whenever it will come out, for example, we'll get the presence of one type of the enzyme that is known as what? 5' mono deiodinase. Deiodinase. It will convert into T3. Now we are getting the T3. Now whatever the thing will choose in the cellular levels. Okay, actually the mechanism will start now. For example, I am just taking a hypothetical cell. Okay, let's take it over here. I think it's not enough space. Anyway, I am just trying to cover these things over here. I am just taking the hypothetical cells over here. So we have taken already what, the T3 and T4, and the T4 is getting converted into T3 in the biologically active form. So we are getting the what, T3. We are getting also the T3 from here. Now see, the T3 is the what? Very small molecule. We can say the single amino acid derivative molecules. So whenever I am saying that the T3 is the single amino acid derivative molecule, that means what? As the size is so less, so they can pass within, through the cell membrane easily. So we'll get the T3, they will be able to pass through the cell membrane easily because see, they are only the what? The derivative of the single amino acid molecule. So here we get actually the the receptors for the T3 that is present within the nucleus of the cell. So whenever we are seeing this T3, okay, whenever we are seeing this T3, they are able to go through the cell membrane because their size is less. So okay, let me over here guys. I'm just making the nucleus of the cell. For example, here is the nucleus of the cell. Okay, let's make it in this way. I think it's enough for you to do all these things over here. Now see, within the nucleus we will see there is a presence of the receptor for the T3. Now if you see the receptors for the T3, whatever the thing we will see, for example I am just showing you guys over here. If we see the T3 receptor, they are having the three parts. For example, this is the first part, okay, this is the second part, and this is the third part. Okay. Now if you see the receptor of the T3, for example, I am writing it over here, T3R. So T3R stands for T3 receptors. So for example, the number 1 portion is the number 2 portion, number 3 portion, number 3 domain. So number 1, if I write it over here, what is number 1? Number 1 is the place where the T3 will get bound. Now if we see within the cells, definitely within the nucleus we will get what, the genetic program. So within the genetic program we will get what, the, okay, let's use this color over here, DNA. So whatever thing we will get over here, try to understand from here. Now see, if we see the structures for the T3 receptors, we will get the first part over here. For example, the T3 is coming over here, and T3 will get bound along with these structures. T3 is getting bounded. Now whenever the T3 will get bounded, it will just push this molecule in these sides. That means what? The T3 that will push the T3 receptors along towards the nucleus. So sorry, towards the what? Genetic program, because in terms of the DNA. Now see, this is the what? The second portions over here. I will write it over here, the name of the second portions of the T3 receptors, that is known as co-stimulator. Co-stimulator. And what about third part? Third part, this part is known as effector domain. Effector domain. Now see, whenever the T3 will get bound along with the first part of the T3 receptors. Okay. Now what is the name of this T3 receptor? Let's write it over here. Okay, let's use this color over here. Okay, let's use this red color only. What is the name of this T3R receptors? That is known as RXR. RXR stands for retinoic acid XR receptors. So what is the name of the T3 receptors? The T3 receptor that stands for RXR, we can say in terms of the retinoic acid X receptors. Okay. Now whenever we will get these receptors will be activated over here, it will the T3 that will push this receptors. That means the T3 whenever it will get bound, it will push the receptors towards the what? Towards the genetic program. We can say in terms of the DNA. Now within the DNA, this is the third part. What is the effector domain? Within the DNA, we will portions. Within the DNA, we will get a portions that is known as hormone sensitive DNA sequence. It is known as hormone responsive. We can say hormone or responsive DNA sequence. So what is this thing that is known as hormone responsive DNA sequence? Now try to understand. Whenever the T3 is getting bound along with the T3R receptor in terms of the retinoic acid X receptor over here, these receptors that will come towards that will go towards the what? That will go towards the this region. That means what? The hormone sensitive DNA sequence. Now the third part, whenever this third part that will get attached along with these portions, after that we will get within the DNA, within the gene, the transcription alternations. We will get the alternations or the changes within the transcription process within the DNA, within the genes over here. So now see, whenever this T3 is just pushing these structures, now let me show the another portions over here. For example, it is now let me show this color with a different color over here. It's the T3, okay, effector domain. Whenever it is pushing, what are the structures will get over here? Again, is the primary stimulator, we can say. Okay, guys. Okay, let's continue. Now see, these are the primary receptors. The T3R, this is the first portion, this is the primary receptors. The second number is the co-receptors or co-stimulator, you can say. Is the primary stimulator, is the co-stimulator. Now whenever the T3 that will push the T3 receptors over here, whatever the thing we'll see over here, try to understand from this portion. This is the we'll get what is the co-receptors, and whatever the thing we'll see over here, see, we'll get this effector domain. This effector domain, see, the effector domain will get bound along with the hormone sensitive DNA sequence. Now see, whenever the T3 is pushing the T3R receptors in terms of the retinoic acid X receptors, whenever it is pushing at that time we'll see the effector domain of the T3 receptors that will get bound along with the hormone sensitive DNA sequence. Now whenever it will get bound along with the hormone sensitive DNA sequence, at that time we'll see alternations in the transcriptions process within the DNA. Now here we get the alternations in the transcriptions. So whenever we get the alternation in transcription, at that time we will get definitely the productions of what? RNA. So first we will get the productions of what? Pre-messenger RNA. This is known as pre-messenger RNA. Pre-messenger RNA. Now whenever the pre-messenger RNA we are getting over here, that will come out of the nucleus. That will come within the cytoplasm, as we know, in the process of translation. So whenever we are getting the mRNA that is coming outside, okay, let me extend some part from here. Okay. I have already said number 3 is the what? Number 3 is the this portion is what? The effector domain. And here what? We will see the binding of the T3. This is the primary stimulator, this is the co-stimulator. Now see, whenever we are getting the mRNA, that will come out. Whenever you are seeing the mRNA that is coming out over here, and we will see this mRNA that will get bound along with the what? Ribosomes, as you know, they will get bound along with the ribosomes. So whenever they will produce what? The different kinds of the amino acids, and as we know that amino acid will get, they will get attached with each other, and as a result we will see the productions of what? Proteins, right? So whatever thing we will see over here. Now we will get over here whenever the mRNA is coming out and they are getting bound along with the ribosomes, as a result we will get the productions of the protein, new proteins. In terms of the new proteins, what are the proteins we will get over here? We will get the functional protein, we will get what? We will get the structural protein, and for example we will get the regulatory protein. What are the functions of each individual protein? Now, so whenever I am talking in terms of the functional protein, try to understand from here. This functional protein, they will what? They will increase the metabolism. So whenever we will see the metabolism that is getting increased, so more amount of the what food is getting breaking down. So at that time we will see that the more amount of the respirations over here, so more utilization of the oxygens. As a result, we will get the more productions of the hot heat. So whenever we will get the productions of the heat, we can say in this way it will just maintain the BMR, right? It will maintain the BMR. Okay, let's write it properly, BMR. Now in terms of the structural protein, what are the functions for the structural proteins over here? Try to understand. This protein that will help for the physical development and mental development, especially in terms of the neurons. Okay, so if we say over here, this is structural proteins that will help in terms of what? The physical development and mental development. And whatever these regulatory proteins, they will activate especially different kinds of the transporters. They will just produce the different kinds of the transporters in terms of the proteins. So what are the transporters? For example, if we see over here, they will increase the amount of the sodium potassium ATPase. So whenever they are increasing the amount of the sodium potassium ATPase, whatever the thing we'll see, so more amount of the this process will be potassium that will be taken, the sodium that will be thrown out. So we'll get this processes. Whenever we see this type of the processes, we'll get the resting protein cells, then the threshold protein cells, we'll get the depolarizations, the different kinds of the activities we'll get within the cells. That is normally supposed to be done over here. So these regulatory proteins that can help over here only. Okay. Now I'll repeat once again these things because this thing is more important. See, as I said, this T3 is having the receptors within the nucleus of the cell. So this T3 that will get bound along with its receptors. So whenever it will get bound along with its receptor, it will just push the T3 receptors in terms of the retinoic acid X receptors. Okay. So as I said, within the receptors will get the three parts. The final part, or you can say the effector part over here, or you can say the effector domain, that will get attached along with the what? Hormone sensitive DNA sequence, or you can say hormone responsive DNA sequence. Now whenever this thing that will get attached along with these portions of the DNA, we'll get the changes in the transcription process, and we'll get the productions of the what? Pre-messenger mRNA. So we'll get the over here we'll get the pre-messenger mRNA. Okay, so whatever the thing, they will come outside of the nucleus, and whatever the thing we'll get, they will get bound along with the ribosomes, and they will produce the new proteins. Whatever the proteins we'll get in terms of the functional proteins, in terms of the structural proteins, in terms of the what? Regulatory protein. Now what are the functions we'll get, I have already explained to you guys. Okay. In the next part we will talk how the BMR is getting regulated. There are also much more mechanisms. Okay, in details I'll just talk about those things. So next part. So thanks for watching. If you think this video is helpful for you, please do like and subscribe my channel, and see you in the next video soon. Okay, and please comment below.